Thanks. Hello everyone. My name is Oindrila Biswas. Today I am going to present my work about how we use the parahydrogen-based polarization method in the reverse Micelles to detect the protein-ligand interactions. Actually, this method can be diversified to be used for biomolecular NMR. Now, we all know that NMR is a widely used spectroscopic technique. However, it suffers from the problem of low sensitivity. But this problem can be overcome by inducing non-Boltzmann nuclear spin state population with the help of hyperpolarization methods such as GNP, FEEP, STABR, CIDNP. So my work is mainly on the FEEP and STABR based methods. So let me just briefly tell this phenomenon. So in FEEP, in an unsaturated substrate, the parahydrogen acts in a pairwise position, and we see the resultant hyperpolarization in either the newly attached hydrogens or the X nuclei which is present in the substrate. In this method, the product is chemically modified, while in Saber-West methods, the product is chemically intact. That is, it is not modified. Here we see the formation of the transient iridium complex, where the polarization gets transferred from the hydride to the substrate, and as a result of the Saber method, we get a hyperpolarized substrate. So both these methods have been used for various applications, such as the hyperpolarized product from the field. They have been used for metabolic imaging and for elucidating catalytic mechanisms and for real-time monitoring of the reaction intermediates. The Saber-based methods, the ligand can be hyperpolarized multiple times, and so this ligands can be used for study of the protein-ligand interactions. So my main aim was to monitor these biomolecular interactions. Now there is one problem for monitoring of this biomolecular interaction. The widely used Saver pre-catalyst, it is not soluble in water, but it is soluble in the organic solvents such as methanol, chloroform. However, the biomacromolecules like protein, they are soluble in water. So now different groups have taken different approaches to solve this biocompatibility issue in such Saber kind of experiments. They have designed and synthesized several water-soluble iridium catalysts. They have used heterogeneous supports to immobilize the catalyst. Also, they have used different solvent mixtures of like methanol, ethanol, along with G2O to tackle this biocompatibility problem. Now we have come up with the approach of using a reverse Micelle, whereby the inside is the aqueous core, whereby the biomacromolecule can be easily solubilized, and the outside consists of the organic phase, where the iridium catalyst, the co-ligand, and the ligand can be solubilized. Also, we can choose a ligand which is soluble, which can like partition in both the organic phase and the aqueous phase. Hence, if the ligand is hyperpolarized in the organic phase, it can traverse to the aqueous phase and transfer the polarization directly to the protein. Additionally, in reverse Micelle systems, as the organic, as it is a, like the water droplets are kind of nanostructures which are in the low volatile organic solvents. Because of this, the correlation time reduces and we can get a better protein spectra. Now, keeping these things in mind, we took the reverse Micelle system with the ABAP ligand. This is because ABAP ligand it consists of the 2,4-diaminopyrimidine moiety, which is a known substrate for the protein which we took, that is the 10A protein, and we took the co-ligand as 4 third-butyl pyridine. Now this is the full spectra of the reverse Micelle solution. Here we can see that the co-ligand proton, it is like enhanced, but we did not see any enhancement for the ligand proton. Here we can also observe that we can see the hydride peaks of the iridium complexes, which are 3 orders of magnitude enhanced. So, as we did not see any ligand enhancement, so we thought of, can we use the transient iridium complex to as a chemosensor to know the concentration of the free ligand concentration in the solution, which in turn can help us determine the protein ligand dissociation constant. So we got 2 sets of hydride peaks. Now to know that which set of hydride peak is for which complex, we took 3 solutions. In one, we took the Saber complex and Saber pre-catalyst and took the co-ligand and found out from the chemical shape that it corresponded to complex 1. And in another solution, we took this saver pre-catalyst and only the ligand and found out the chemical shape of the complexes, which corresponded to complex 3. In the third solution, we took the ligand as well as the co-ligand and the saver pre-catalyst, and we got three sets of hydride peaks, which corresponded to complex 1, 2, and 3. So thereby the two hydride peaks which we got, they are from complex 1 and complex 2. We also found out that 20 minutes was required for the full activation of the catalyst to form complex 1 and complex 2. Now we did the reaction in absence of protein. In when we solubilized the protein and made the reverse Micelles solution, we found out that in case of for complex 1, the intensity of the signal in absence of protein and in the presence of protein, they are somewhat similar in intensity. However, for complex 2, the intensity decreases in presence of protein, which is due to the fact that in presence of protein, the ligand binds with the protein, and as a result of which we see a less in concentration of free ligand, and complex 2 also forms less in concentration. So if we define RH as the ratio of ratio of intensity of complex 2 to that of complex 1, then this RH value decreases very much on addition of protein in the reverse Micelles solution. So which this decrease in RH value we can use for determining the dissociation constant of the protein ligand. Now we see that in the reverse Micelles solution, there are like three equilibriums. First, the IRC2 complex, that is complex 1 and complex 2, they are in reversible equilibrium. So from this, using the RH value in absence of the protein, we can determine the concentrations of these components and also the K equilibrium value. Now the ligand is in like partitioning between the aqueous phase and the organic phase, and from this equilibrium we can know the free ligand concentration in the aqueous solution. Also, the ligand is reversibly binding with the protein in the aqueous phase, and by we know the free ligand concentration in the aqueous solution. So from here we can determine the protein ligand dissociation constant. Also, this value agrees very well with the dissociation constant, which we have determined independently by with the help of the Michaelis-Menten equation. Now we want to see that we the previous experiments we have used like 100 micromolar of ligand concentration. We want to see that if we use a lower ligand concentration, are we still able to see the complex 1 and complex 2 peaks or not? So here we used 1 micromolar of ligand concentration, and we saw that still we 1 and complex 2 peaks are observable. Now we did all the reactions with using just one scan. This if we increase the number of scans here, because in the reverse Micelles system it is a like one pot system, so we are just mixing everything, and in the one pot it is like after vortexing the thing, the reverse Micelle is ready. So the reaction can be repeated many number of times, and we can also hence increase the number of scans, which will increase the signal to noise ratio. Thereby we can also operate at a much lower ligand concentration. The previously used ligand, we did not see any direct hyperpolarization of the ligand, but we wanted to see, like, if we use other ligands, can we see the hyperpolarization, direct hyperpolarization of the ligand in this reverse Micelles system or not? So we took nicotinamide, 2,4-diaminopyrimidine, and 3-aminopyridine, and with different co-ligands like DMSO and acetonitrile. We observed the saver enhancements in this reverse Micelles solutions only. So the ligands were directly hyperpolarized in this reverse Micelles solutions. So we also checked the like partition coefficient of the saber catalyst, and we found out that it is only soluble in the organic system. So the nature of hyperpolarization can be happening such that the ligand which we took are all soluble in both the organic phase and the aqueous phase. So initially, when the ligand is in the organic phase, it is getting hyperpolarized and the iridium complex is forming. Then the hyperpolarization is being transferred to the ligands directly, and the ligand, if it goes to the aqueous phase, it can directly hyperpolarize the protein also. So we developed the method in which we can do the reactions in one pot and directly hyperpolarize directly the ligand in the reverse Micelle solution. So, in summary, we have used the reverse Micelle to solve the biocompatibility issue in this kind of saber experiments. Also, as in the first case, our ligand was not hyperpolarized directly. We used the iridium complex as the chemosensor for knowing the free ligand concentration in the solution and also to determine the protein ligand binding affinity. Later, we were also able to directly polarize different ligands. Now, in the first case, as there was a bulky ligand, it did not polarize directly. So it may also happen that that ligand, the ABAP ligand, because of its bulky nature, it bound to the iridium complex through the axial position and not through the equatorial position. That's why we couldn't see the direct hyperpolarization. However, as these ligands they are smaller in nature, they bound to the iridium complex through the equatorial position and hence were like hyperpolarized directly. So I want to thank Dr. Pierce and Professor Christian Hilty for immensely like contributing to the projects. Also, I want to thank the funding agencies and like all of my group members for maintaining such positive environment in the lab. Thank you.
Thank you, Oindrila, for a very interesting talk. If there are questions, please type them into the Q&A. And are there questions from the room? So I have one. So for compound 2, you showed a sensitivity that was like 120 nanomolar, something like that, right? In terms of your sensitivity.
Yeah,
It's 2. Yeah, you just passed it. I don't know
yeah,
where, but it was super impressive. Yeah, that's it. So yeah,
yeah,
120 nanomolar. So
yeah.
ultimately, what is setting that sensitivity? Is it just basically like, okay, of course there's the system noise floor, but is it just essentially it's the enhancement that's attainable, or is it something more sort of fundamental than that? Binding efficient. Like, do you think that that's basically the limit of where we're going to get, where you're going to get with this approach? It's a very amorphous question.
I,
Let me be more clear. Can you comment on that very impressive sensitivity of 120 nanomolar and where you might expect the kind of detection limit ultimately will end up? Maybe that's around where it's going to end up.
I haven't checked a concentration lower than that, but here the hydride signals they were like 3 orders of magnitude enhanced, and that's why we could see like such a lower concentration of the complex two, which was observed in this case.
Mm-hmm. Great. Any questions from the room on our side? See any questions in the chat? So let's thank our speaker again. So the next speaker in this promoted talk session is Sergey.
Hello, dear colleagues. Do you hear me?
We are not really getting your audio, Sergey.
Hello.
Okay, that's better.
Better. Okay. Do you see my screen?